Hi, this is a recording to accompany the slides for the first out of two lectures on the anatomy of the nervous system. My name is Dr. Alita Partosudarso, and I'll be walking you through the slides. So to begin with, I'm going to talk about what I'm going to cover in this lecture. So the first one is talking about the various ways you can actually divide the nervous system, followed by a brief discussion on the coverings of the neurons in the peripheral nervous system and the definition and location of various ganglia around the body, and then going to the central nervous system and again talking about the various coverings that surround the neurons in the central nervous system, and a brief discussion on the blood flow within the central nervous system, as well as the production, flow, and resorption of cerebrospinal fluid. So to start off with, the nervous system can be divided according to location. And when we divide the nervous system into its location, we can divide it into the central nervous system and the peripheral nervous system. So if you see over here, the peripheral nervous system, the central nervous system consists of the brain and spinal cord. So you've got here the brain and spinal cord, and the brain and spinal, within each of the brain and spinal cord, it is divided into visually distinct region. And the visually distinct region is dependent on the color of each of the regions. It can be divided into a region of gray matter and a region of white matter. And the reason for the visual division within the central nervous system is that they're parts of the brain and spinal cord that look paler or more white than the other parts. So the parts that are paler are known as the white matter. The parts that are not so pale are known as the gray matter. In the spinal cord, as you can see over here, white matter is located superficially compared to gray matter. So if you look over here, the white matter is on the outside of the spinal cord and the gray matter is on the inside of the spinal cord. So the gray matter is located deep, white matter is located superficially. In the brain, as you can see over here, the white matter is located deeper compared to gray matter. So you can see here white matter on the inside deeper, gray matter on the outside more superficially. In the central nervous system, regions that contain mainly cell bodies and dendrites are called the nucleus and these are present in the gray matter. And the regions that contain mainly axons and myelinated axons at that are called tracts. And these are also referred to white matter. So that's the central overview of the central nervous system. In the peripheral nervous system, it contains neurons that are not found in the central nervous system, right? So everything that's not in the brain and not in the spinal cord. And in the peripheral nervous system, regions that contain mainly cell bodies and dendrites are called ganglia, or ganglion for singular. And regions that contain mainly axons are called nerves. And we're going to talk about that as we move along. And so I just wanted to bring to your attention this diagram over here of the eye and the nerve or tract. So you can see here where the neuronal axon is in the peripheral nervous system is referred to as the optic tract. And when it enters into the central nervous system, in the peripheral nervous system, it's called optic nerve. And when it enters into the central nervous system, it's referred to as the optic tract, right? So axons in the central nervous system called tracts, axons in the peripheral nervous system are called nerves. So this is one way to divide the nervous system. Another way to divide the nervous system is according to the effectors. So the effectors are the cells and tissues and organs that are responsible for doing the bidding of the nervous system. And so if we divide it according to the effectors, we can divide it into the somatic nervous system over here and the autonomic nervous system. When we look at the somatic nervous system, the effector tissue that is connected to the somatic nervous system are the skeletal muscle. So skeletal muscles are voluntary muscles, and they're responsible for conscious perception and voluntary motor responses. So that's the somatic nervous system. The autonomic nervous system is this box over here. So the autonomic nervous system has got 3 divisions. The autonomic nervous system is called autonomic because it's automatic. So the control of the autonomic nervous system is not under our voluntary control, which is awesome because we don't have to think about how fast or how hard our heart should beat. We don't have to think about digesting all the food that we've eaten. We don't have to think about a whole lot of things because that's all automated, right? That's all part of the autonomic nervous system. Autonomic nervous system is divided into 3 different parts. So the first 2 parts are the sympathetic and the parasympathetic divisions. The parasympathetic divisions, as it says over here, conserves energy. So the parasympathetic division is what is most active when we're not doing much, when we're sitting, when we're reading, when we're watching Netflix, when we're thumbing through our social media. Promotes housekeeping when we're sleeping, promotes housekeeping functions. The other division, the sympathetic division of the autonomic nervous system, is involved in the fight, flight, or freeze responses. So this is the nervous system that kicks in when we're challenged, right? When we're nervous, when we're excited, when we meet a bear, when we don't quite know what to do. It's the fight or flight reflex. And then the third division is called the enteric nervous system, right over here. Enteric refers to intestinal, so it's found entirely within the walls of the digestive tract or the gut. The enteric nervous system can function independently of the central nervous system. So if you were to take a piece of stomach out, a piece of small intestine, a piece of large intestine out, it can actually function on its own without any input or input from the central nervous system. So the effector tissue of the enteric nervous system is the smooth muscle and the glandular tissue within the digestive tract. So this is another way of dividing the nervous system. A third way of dividing the nervous system is according to the direction of information flow. So you've seen this before, and what I can say is I'm going to repeat it again. So again, if you divide it according to the direction of information flow, it really is split into the sensory or afferent division that sends information up to the central nervous system, and the motor or efferent division that receives information from the central nervous system. So again, this is the whole neuronal arc where you have to start off with, you have a stimulus, which activates a sensor, right? A stimulus is any change from homeostasis, whether the change is generated internally or externally. Information is sent via the sensory or afferent neuron, and afferent means towards, in this case, towards the central nervous system. In the central nervous system, there's a lot of different inputs that go into the central nervous system. Central nervous system then integrates all the inputs and generates an output, and that processing and integration is done by interneurons within the central nervous system. And then what happens is it generates a response. So once the response is generated, it's sent to the periphery via the motor or efferent neuron. And efferent means away. In this case, the information is sent away from the central nervous system. And if you look over here, then what happens is it sends it to either the somatic nervous system or the autonomic nervous system, in which case then the response is generated either voluntarily with conscious perception and control, or involuntarily without any conscious perception or control. Just as an overview of an example of how the nervous system works, this is what happens whenever you take a shower, right? So when you take a shower, you turn on the tap, you put your hand underneath the water that's coming out of the shower, and you have sensory nerve endings in the skin, which detects a change in temperature, whether the water or a change in input, whether the water be hot or cold or lukewarm. And that stimulates the sensory endings. And if the sensory endings are appropriately stimulated, then threshold is reached, action potential is sent via the sensory neuron. And what happens is that the action potential travels via the sensory neuron all the way to the spinal cord, where it synapses or makes a connection with a second neuron. Then the second neuron sends a signal again up towards the central nervous system. More often than not, it reaches the thalamus. Then what happens is the thalamus then sends generates a signal in the third sensory neuron, which projects up to the cerebral cortex. And once it reaches the cerebral cortex, then integration happens and processing via the interneuron, and the response is then sent down motor neurons. So you've got upper motor neurons that starts from the central nervous system and goes out of the central nervous system, and it synapses with the lower motor neuron over here in the spinal cord, and the lower motor neuron then sends a signal out to the hand to see whether you want to jerk the hand away from the hot water or you want to adjust the temperature depending on the sensation generated by the sensory stimulus in the first place. I'm going to move on and talk about a couple of slides on the peripheral nervous system. So the peripheral nervous system are organized. So what happens is that you've got these nerves, which if you actually look at a nerve, you can see that they've got a lot of different neuronal axons or fibers, right? So each of these long things is an axon of a nerve. And when you have bundles of things, what happens is you have coverings, protective coverings, connective tissue coverings around these bundles, right? So again, in similar style to the coverings around muscle fibers of skeletal muscles, nerves have a similar, peripheral nerve axons have got a similar organization to their connective tissue coverings. So the first connective tissue covering of the nerve is called the endoneurium. So neurium refers to nerves, and the first part refers to its location. So the endo is inside. So these are the inner covering, most delicate, that covers each nerve axon. So each of these axon over here have got an endoneurium covering it. And those nerve axons are then bundled into fascicles, right? Similar to muscle fascicles. So what happens is you've got endoneurium covering each nerve axon, and then they're bundled into these fascicles. And each fascicle has got a perineurium covering it. So you've got a perineurium covering that, you've got another one covering that. So each of these are covered by a perineurium, right? So this is an axon, so you've got endoneurium over here, and you've got here lots of different axons forming a fascicle, and then you've got a perineurium covering the entire single fascicle. And what you can see here is that this nerve has got several different fascicles, and covering the entire nerve is a stronger outer coat called the epineurium. So endo, smallest, each individual nerve axon, um, peri covering fascicles of axons, and then the epineurium covering the entire nerve. Nerves are associated with a CNS region that they're connected to. So basically, cranial nerves are nerves that project outside of the CNS that are connected to the brain, the cranium, whereas spinal nerves are nerves that project outside of the central nervous system that are connected to the spinal cord. So these are the axons, right? So if you look at the nerve, these are this portion over here. And the next slide basically talks about the ganglia. So a ganglia, like the way that nerve axons are bundled together, ganglia are a collection of cell bodies that are grouped together. So you've got, all right, a ganglia would be, oh, I can't draw. One more. So the ganglia would be this collection of cell bodies. And the dendrites of the cell bodies, as well as the synapses between the dendrites of the nerves inside the ganglion and the nerves that project to the ganglion. So you've got a lot of different synapses that occur within a ganglion, and the action potential travels from the end of the axon terminals of these blue nerves to the dendrites of the red nerves, right? So in terms, the the ganglia are organized according to location as well as function. So we've got the dorsal root or posterior root ganglia that are associated with spinal nerves. So spinal nerves are peripheral nerves that come out of the spinal cord, right? So this is the spinal cord. This is the spinal nerve over here. And the dorsal root ganglia is an enlargement in the dorsal root that contains cell bodies and dendrites and synapses. Or just cell bodies in this case. Cranial nerve ganglions are ganglions that are associated with a cranial nerve. So these in the green are the cranial nerves here. Sympathetic chain ganglia are associated with sympathetic chain, paravertebral, and prevertebral ganglia are ganglia. They're associated with the sympathetic nervous system that we're going to look at later on. So these are the sympathetic chain ganglia. And they're called sympathetic because they're associated with the sympathetic nervous system. And they're called chain ganglia because as you look over here, they form a chain, right? So sympathetic chain ganglia are indicated here. And then you've also got other ganglia associated with a sympathetic nervous system, which is the paravertebral and the prevertebral ganglia. And again, we're going to look at these ganglia when we talk about the autonomic nervous system. The other part of the autonomic nervous system is the parasympathetic nervous system. And in the parasympathetic nervous system, there are terminal ganglia associated with it. And it's called terminal because these ganglia, this collection of cell bodies, are located either very close or in the walls or tissue of the target organ that is associated with the parasympathetic nervous system. So you can see here, so the parasympathetic nervous system starts in the CNS, projects outwards, and it ends in the terminal ganglia that are located right in the organ walls that it innervates. When we look at the central nervous system, what we can see is that, again, they have coverings. However, in the central nervous system, the coverings are around the entire brain and the entire spinal cord. These coverings are called meninges. Again, there are three different types. The inner one is the most transparent, most delicate one. It's called the pia mater right here. And then the second one is called the arachnoid mater because someone decided that it looked like a spider web, so they called it the arachnoid mater. Not quite as delicate as the pia mater. And then the last one is the dura mater, which is durable, right? Which is strong, outer, fibrous, connective tissue membrane that surrounds the entire brain and surrounds the entire length of the spinal cord, right? So the dura mater is on the outside, arachnoid mater in the middle, pia mater on the inside. In between each of these things, there are spaces. So you've got the subdural space, that is, sub means under, so the subdural space is under the dura mater, and so it's located between the dura mater and the arachnoid mater. And then you've got the subarachnoid space that is sub under the arachnoid mater, right? And then you've got the epidural space, which is not depicted over here, which is located between the dura mater and the spinal cord and vertebrae, and it contains fat. And we'll look at that when we talk about the central nervous system in more detail in another slide. So when you look at this, what you can see is you've got three different connective tissue lining. What I want you to see is the dura mater. The dura mater also extends deep into different parts of the brain and spinal cord. So we have these extensions of the meninges, right? So in the brain, we've got the dura mater, which has extensions to separate different parts of the brain. So you've got within the subdural spaces, you've got the dural sinus, which functions as veins. So it collects blood in the central nervous system in order to return it to the heart. You've got an extension of the dura mater, which forms what's called the falx cerebri. So it extends deep within the cerebrum to separate out the right and left cerebral hemispheres. You've got the same thing happening in the cerebellum, which extends in the medial section of the cerebellum to separate out the right and left cerebellar hemispheres. And you've also got another extension of the dura mater in between the cerebrum over here and the cerebellum over here. So it separates out the cerebrum from the cerebellum, and this is called the tentorium cerebelli. Right? So each of these dura mater extensions into the brain basically separates out the brain into its various large components. In the spinal cord, we've got the innermost meninges, the pia mater, having extensions. And the extensions of the pia mater actually helps to anchor the spinal cord so that your spinal cord doesn't bounce around when you walk, jump, run, skip, whatever, when you move, right? So it helps to anchor the spinal cord within the vertebral foramen of the various vertebrae that you have. So you've got the denticulate ligaments, which extends outwards from each section of the spinal cord, and it basically anchors the spinal cord to the vertebrae sideways, right? Transversely. And you've got another extension of the pia mater right at the end of it called the filum terminale. So this filum terminale extends from the end of the spinal cord and it basically anchors the spinal cord to the coccyx. So with these two extensions, you have the spinal cord over here, you've got the filum terminale, and then you've got the denticulate ligaments. These basically anchor the spinal cord so it doesn't bounce around and doesn't sort of wiggle up and down and move side to side when we're actually living life. The next thing I'm going to talk about is the circulation of blood in the brain. So the first one is on the arterial circulation, right? The provision of blood to the brain from the heart. So because the brain is a protected organ, we need to ensure a constant, consistent supply of blood to the brain. So we've got a whole bunch of arteries that supply blood to the brain. We've got the carotid arteries, right and left, common carotid arteries that come directly from the aorta and also contain some receptors to monitor the chemical composition of blood as well as the blood pressure in the carotid arteries. You've got the vertebral arteries, which travel within the transverse foramen of the cervical vertebrae and enter the cranium through the foramen magnum of the occipital bone, and it merges into the anterior spinal artery and the basilar artery. And we've also got this thing called the circle of Willis here. And the circle of Willis ensures that we have as much as possible continued blood supply to the brain. So if you look at the circle of Willis over here, what you can see is you've got a lot of different arteries feeding into the circle of Willis. So what this means is that if any one of these arteries are blocked, blood can still circulate into the brain with the help of other arteries that are not blocked. So the circle of Willis, very important to maintain consistent blood flow to the brain. In terms of returning blood from the brain to the heart, we've got the dural sinuses. So you can see the dural sinuses is an empty space that the veins in the central nervous system drain into. And it also also collects the cerebrospinal fluid from the meninges. And what happens is all the blood and the cerebrospinal fluid travels along the sinuses, right? Superior sagittal sinus is found right in the medial section of the cerebrum, and then it travels through these various sinuses into the jugular veins, and from the jugular veins it makes its way back to the heart. Speaking of cerebral spinal fluid, this is the last section of this lecture. So cerebral spinal fluid is produced from blood, from the filtration of blood through these ependymal cells, which are found lining the empty spaces in the brain called ventricles. So this is what happens. So what you have is you have the functions of cerebrospinal fluid. So it helps to remove waste from the interstitial fluids of nervous tissue and return them to brain. It also helps, if you look at the location of the cerebrospinal fluid, you can see in the dark blue, it helps to provide a protective, supportive cushion around both the brain and the spinal cord. And it's also a reservoir of circulating fluid. And it also has some receptors, which then detect chemoreceptors, which then detect changes in the chemical composition of what's going on in the central nervous system. As I said, cerebrospinal fluid is made by the filtration of blood through the sheets of ependymal cells. So if you look over here, you've got these ventricles. So ventricles are basically open spaces in the brain that contain these choroid plexus. And the choroid plexus is lined with ependymal cells. And so the blood in the network of capillaries in the choroid plexus is filtered through the ependymal cells to produce cerebrospinal fluid. So the main difference between cerebrospinal fluid and blood is that it contains no cells. So there's no white blood cells or red blood cells in cerebrals or platelets in cerebrospinal fluid, whereas all those cells are found in blood. In terms of where CSF is generated and where it travels, so as I said, it is generated in the choroid plexus of the ventricles. And what happens is you've got the ventricles, the lateral ventricles. You've got right and left lateral ventricles in the cerebrum, in the top part of the brain. Is where some CSF is formed, and it travels through the interventricular foramen. So foramen means hole, interventricular means in between ventricles. So right and left is the first and second ventricles, and then it goes through the third ventricle, which is in the deeper part of the brain, in the diencephalon, through to the cerebral aqueduct, right? In the midbrain, which is part of the brainstem, and from the cerebral aqueduct, it travels through the fourth ventricle in the brainstem, and at that time it splits in two directions. So first of all is that it goes down through the central canal of the spinal cord, and the second one is it goes into the subarachnoid space of the brain. And in the subarachnoid space in the brain, you've got cerebrospinal fluid being returned into the venous blood supply through the arachnoid villi in one of the sinuses in the collection of blood that exist in the brain. And that's it. I hope you've enjoyed it, and I'll see you next time.